What would you put in your magic anti-aging pill? Most certainly a molecule that promotes longevity and that has the ability to delay the development of one or more age-related diseases. One able to improve the human biomarkers of aging, to reproduce its effects in different model organisms, and that has minimal side effects. A molecule targeting the mitochondrial function, one of the main players in aging, would be fantastic, and even more so if it works on autophagy and mitophagy, as well as on muscular function. Imagine a molecule that is proven to be effective not only in mice, but also in humans. Recent evidence shows that there is a pleiotropic molecule that can develop multiple beneficial effects on cardiovascular, neurodegenerative, and metabolic diseases, on cancer, but also on muscle mass, sarcopenia, osteoporosis, osteoarthritis, vitamin D, diabetes, and more. But our patients also require more from us. They want a supplement that it's easy to understand, easy to buy, that works, that increases their performance, is not too expensive, doesn't require daily injections, and is safe. There is a geroprotector that satisfies all these requirements in a experimental model, and for which there is an increasing interest for its potential application in regenerative medicine as a nutraceutical tool. It is known urolithin A. It's a natural metabolite produced by gut bacteria from ellagic acids and improves health and life expectancy in various experimental models. So, I investigate urolithin A, analyzing the mechanism of action with particular regards to mitophagy, the studies demonstrating its efficacy and safety, and its application in clinical practice. I would like to thank the organization and the director of Regional Research Group for the invitation. Urolithin A is a natural metabolite produced by gut bacteria from ellagic acids, a polyphenol found in various plant-based foods such as berries, nuts, and pomegranates. In the gut, ellagic acid is converted by the gut microbiota into metabolites called urolithins. We can observe that there are various types of urolithins. Urolithin A is the one with the most healthy properties. Urolithin production depends on individual's gut microbiota, called the metabotype, which defines the individual's ability to produce specific metabolites from food precursors depending on the metabolic profile of the gut microbiota. Three main urolithin metabotypes have been identified: metabotype A, urolithin A producers; metabotype B, and metabotype 0, urolithin non-producers. So, after eating foods rich in ellagic acids, only a portion of population is able to efficiently convert these compounds into active urolithin A. And those who do have better metabolic health parameters and microbiota composition of the others. Of course, direct supplementation with urolithin A bypasses the need of microbial conversion. The capacity to produce urolithin A decreases with age, from 61% in 20 to 50 years old to only 39% in those over 60. The mechanisms of action are multiple, even if the activation of mitophagy is the main one. Mitophagy is mediated by two main pathways, the PINK1 parking dependent pathway and the PINK1 parking independent pathway. The PINK1 parking dependent pathway is based on phosphorylation of parking by PINK1, a protein kinase that accumulates on the outer mitochondrial membrane of dense mitochondria, leading to the elimination of these mitochondria. Urolithin A is able to increase the levels of these two key proteins, thus promoting the turnover of dysfunctional mitochondria. The PINK1 parking independent pathway involves mitochondrial receptors that directly bind SC3 and trigger mitophagy. There are also other mechanisms. Urolithin A activates AMPK, inhibits inhibits mTOR, the physiological inhibitor of mitophagy, increases PGC1 alpha levels, and thus improves mitochondrial biogenesis. It activates SIRT1 and FOXO with antioxidant and anti-inflammatory effects. It reduces inflammation by inhibiting NF-κB and pro-inflammatory cytokines and activating NRF2. It has the ability to modulate oxidative stress either by enhancing endogenous antioxidant defenses or by inhibiting the production of reactive oxygen species. Studies has shown its capacity to enhance mitophagy by activating the expression of glutathione S transferases. Urolithin stimulates the NRF2 signaling pathway, subsequently upregulating the expression of glutathione S transferases. At the same time of the stimulation of the mitochondrial cleanliness, it also improves key functional parameters such as respiratory capacity, leading to an overall improvement in mitochondrial function. But there is more than this. In an interesting article published in Neuroscience, it was shown that urolithin A increases clotting levels and promotes longevity in mice. The study was conducted on wild-type mice and mice genetically modified to have low levels of clot2. Urolithin A was administered orally for 12 weeks, and clotting levels, markers of aging, and longevity were measured. Urolithin A significantly increased serum and tissue clot levels and clotogen expression in the kidneys, and it increased the mean survival by 18%. Of course, additional clinical studies are needed to confirm these effects in humans, but it's interesting. The positive effects on aging mediated by mitophagy is conserved from worms to mammals. It extends lifespan and improves half-span parameters such as muscular function. Let's analyze the pleiotropic effects. Urolithin A is negatively correlated with total and LDL cholesterol, meta-inflammation, and BMI, and has a positive potential on cardiometabolic health. It prevents damage and reduces ischemic damage and atherosclerosis in animal models. It improves insulin sensitivity actively on mitochondrial function in skeletal muscle. This article, published in PLOS Biology, shows that urolithin A exerts anti-obesity effect through enhancing adipose tissue thermogenesis. Urolithin A treatment increases energy expenditure by enhancing thermogenesis in brown adipose tissue and inducing browning in white adipose tissue. Urolithin A increases thermogenesis is favored by an elevation of the thyroid hormone liothyronine, T3, in the brown adipose tissue. Consistent with this mechanism, urolithin loses its beneficial effects on brown adipose tissue metabolism when thyroid hormone production is blocked by an inhibitor such as propylturacil. Conversely, administration of exogenous theta-iodothyronine to propylturacil-treated mice restores urolithin-induced activation of brown adipose tissue and metabolism. Urolithin supplementation has also shown promise as a treatment of senile osteoporosis. It significantly reduced the expression of osteoclast-related genes and bone resorption. In an experimental model, urolithin A inhibited bone loss in the proximal femur, spine, and tibia. It even develops positive effects on intervertebral disc degeneration and osteoarthritis by the same mitophagic mechanism. This is an exciting study published by Sarah Livingston of the Arizona University. They show how urolithin A and vitamin D cooperate to amplify serotonin gene expression in neuroendocrine cells. Urolithin enhances the ability of vitamin D to induce the serotonergic enzyme in tryptophan metabolism to serotonin, namely TPH2, and function as a booster of the action of vitamin D to stimulate gene expression via VDR. Leptin normally inhibits synthesis and release of brain-derived serotonin. Vitamin D represses adipocyte leptin and induces TPH2 to enhance serotonin release signaling in the cerebral cortex. So a combination of vitamin D and urolithin A is an attractive potential to raise serotonin in the central nervous system and perhaps improve mood. Skin aging. Research shows that urolithin A significantly increases type 1 collagen expression, reduces matrix metalloprotease 1, reduces intracellular reactive oxygen species in certain human skin fibroblasts. It exerts protective effects against UVA-induced photoaging and prevented various morphological changes typically observed after UVA exposure through NRF2 activation and mitophagy. Neurodegenerative diseases. In neurons of patients with Alzheimer's disease, mitophagy is deficient, leading to the accumulation of malfunctioning mitochondria. Urolithin A reduces beta-amyloid and hyperphosphorylated tau protein by activating mitophagy and SIRT1. It attenuates neuroinflammation, oxidative stress, and neuronal damage, and prevents cognitive decline. So, the beneficial effects of urolithin A on brain function are associated with a multi-target action that include promoting mitophagy and mitochondrial function, attenuation of oxidative stress and inflammation, inhibiting beta-amyloid and tau protein, and regulating tryptophan metabolism, thereby counteracting the pathogenesis of Alzheimer's disease. Urolithin A is also known for its anti-cancer properties. I mentioned these trials that investigate the impact of urolithin A on colorectal cancer cells. The authors conclude that urolithin A decreases cancer cell proliferation, delays cell migration, and potentially prevents metastasis. In these other papers, the authors demonstrated that urolithin A inhibits androgen prostate cancer cell growth proliferation. And finally, many findings established a solid scientific foundation for the application of urolithin A in improving muscle health, promoting muscle growth, and enhancing exercise performance. Urolithin A may offer potential benefits in the treatment of muscle atrophy in pathological conditions, but also sarcopenia or extended periods of immobility. It can stimulate muscle protein synthesis while inhibiting protein degradation pathways, helping to preserve muscle mass and mitigate muscle wasting. It can stimulate muscle protein synthesis, promoting to muscle growth and maintenance. These effects are particularly beneficial for individuals looking to build and preserve muscle mass, such as athletes and fitness enthusiasts. It shows promise in delaying the onset of muscle fatigue during prolonged or high-intensity exercise. By enhancing mitochondrial function in muscle cells, it provides sustained energy support, allowing individuals to engage in longer and more intense training sessions. By suppressing the inflammatory response, it contributes to the mitigation of muscle pain and inflammation and promotes muscle repair and recovery. How urolithin acts on muscle health and performance? Professor Zhao gives some such suggestions in these recent papers. Urolithin A has been shown to activate AMPK, to inhibit NF-κB, and enhance the expression and activation of PGC1 alpha, leading to improved muscle health and performance. It has the ability to enhance muscle protein synthesis and degradation by suppressing FOXO and by inhibiting ubiquitin proteasome system and mTOR. Moving on to human studies. The first human clinical trial with urolithin A administration was a randomized double-blind study conducted over 4 weeks in healthy elderly men and women aged 61-85 years. Urolithin A showed a favorable safety profile with no observed side effects, both after a single oral dose up to 2,000 milligrams or after multiple doses up to 1,000 milligrams, with good oral bioavailability at all doses tested, and this is important. This is a very famous study, randomized, double-blind, placebo-controlled clinical trial conducted on 88 overweight and sedentary middle-aged adults, divided into 3 groups: placebo, 500, and 1,000 milligrams of urolithin A for 4 months. Urolithin A at both dosages significantly improved lower limb muscle strength, and it clearly showed the involvement of all the pathways we discussed earlier. Mass proteomic analysis reveal activation of pathways related to mitochondrial metabolism and mitophagy, increased levels of active phosphorylated form of parkin, increased mitochondria DNA, nuclear DNA ratio as an indicator of mitochondrial content, increased respiratory chain protein in skeletal muscle, a significant reduction of plasma C-reactive protein. This is another randomized, double-blind, placebo-controlled clinical trial conducted in older adults. It's interesting because it showed an improvement in the endurance of both hand and leg skeletal muscles, hence a muscle that anatomically and functionally are different. Urolithin has emerged as a multifunctional molecule against etiopathogenetic factors common to multiple age-related diseases and aging. It's not a magic pill, but it is a smart and multi-target pill. It can be used alone or in combination with other mitophagy activators, such as spermidine, resveratrol, NAD, or NAD booster with a synergistic effect, in addition, of course, to physical activity and intermittent fasting. What are the indications and dosages? It can be used as anti-aging compound, of course, for the prevention of cardiovascular, neurodegenerative, and metabolic diseases, for energy and muscle health, for individuals that have no time or interest or physical activity was sarcopenic, but also for individuals that enjoy physical activity and want to enhance their performance, muscle growth, maintenance, and recovery. Studies report dosages of 1,000 to 2,000 milligrams per day. 250 milligrams twice a day may be enough. So, what have I done after studying all this research paper? I took urolithin myself, of course, and appreciate its beneficial effects. Thank you for your attention.